Greetings, my name's Logan Hill. I'm a part of the Freshman Research Scholars Program here at Oklahoma State. I originally came in majoring in biochemistry, but I've recently changed to microbiology due to my experiences in the lab. So the Freshman Research Scholars Program has been very helpful for me. And now I get to present my research that I've just begun working on. So this project is called Fast Growing Synechococcus, and usually we actually call it lab-based evolution in the lab. So really it goes by both names. But I'm a member of the Burnup Laboratory. That's my home for research here, and I'm pretty excited to talk about this project. So let's get right into it. All right. So to introduce it, Synechococcus 7942 is the genus and species of the subject of my experiment. It's a cyanobacteria, which is the phylum that it originates from. This phylum cyanobacteria is full of photosynthetic organisms. They conduct photosynthesis in order to produce their energy. And so we use them to study photosynthesis since they're much easier to use than plants. Um, while they are much easier to use in plants, one of the problems with them is that they can take a long time to grow compared to other bacterias. And this can really hinder a lot of our experiments, especially Synechococcus, which can take up to 6.5 hours to double itself. So it's a challenge. But luckily, with the help of other research laboratories across the United States and a little bit of our own intuition, we've been able to improvise an innovative approach to this problem. Um, we call this project lab-based evolution, and the goal is to influence random mutations so that we can produce a mutated Synechococcus 7942 species that will grow faster thanks to its genetic changes. And then in the future, we might want to know what exactly these genetic changes are and how they affect how each one individually affects the development of Synechococcus 7942. And that has a couple implications that we'll get into in a moment, but that's the beginning. So why is this important? Well, first off, we need quicker laboratory experiments. That's always a bonus so that we can obviously do more research with the time we have. If we're successful in our methods could be applied to other research laboratories and other bacteria as well, because a lot of our methods for doing this are more nuanced and adding on to what knowledge we already know and what studies that have already been published, which we'll get into. A multifaceted approach maximizes our potential changes, which basically just means our methods could be adopted by other labs or by ourselves in order to approach other species that we study for photosynthesis, such as Seneca cystis, which we often use. And also building onto the knowledge of fast-growing cyanobacteria, which is basically at the end, when we find the genome of our species and figure out what's changed, what that affects, and how exactly we've improved the Synechococcus species. So the actual plan for our research is to use Synechococcus 7942 and fill multiple flasks with media. We want 6 separate populations exactly. These populations will have no interlap. They'll mutate and change on their own, irrespective of each other. They'll be grown in prime conditions, which is high CO2 concentration, lots of light, and prime temperature conditions, which is about 30 degrees Celsius. And then about every other day, we'll take a small amount of that solution. We'll take it from a specific place. We haven't decided on a place yet because we're not exactly sure where the most fit bacteria settle at, whether they settle at the top or the bottom. So we'll probably conduct a couple experiments to figure that out as well. But we'll take a small amount of the most fit bacteria in the solution, and then we'll move them to another solution and let them keep growing again. So we're trying to artificially select for the most fit bacteria in order to eventually produce an extremely fit sample. And as you can see in our figure over here on the right side, um, the photo on the left and photo on the right would be in the given same time period. So if, pre our lab force evolution, in one day, maybe our solution gets that thick. And then on the right side, in one day after our evolutions, it gets that much thicker, to put it non-academically. But yeah, then what's left of that will be tossed, and the new population will be allowed to reproduce. and we'll keep doing this process for probably over a year with constant monitoring and going through it. At the end of each week, we'll take that week's sample and we'll throw it on a plate, and then we'll freeze it, and it'll act as a save point in case we get an infected sample, and we can use it to compare eventually with either earlier samples or later samples as well. So it's sort of like saving points. So yeah, it's it was adopted from the Linsky lab, which was a big influence on our laboratory research, which we'll talk about in a little bit. So, what potential results could we expect from this experiment? Well, luckily for us, the results of each of our experiments will be tested constantly thanks to our multi-cultivator, which we'll get into more in a moment. But basically, if you look at the Pakrasi study from 2018, they were able to achieve a 66% reduction in the doubling time of cyanobacteria. Usually, it took 6.8 hours in order for Synechococcus 7942 to double. They were able to get that time down to 2.3 hours. 6.8 to 2.3, 66% reduction. Really amazing results. And so, how we plan to exactly observe our results is to test each population's light absorbance over the same growth period. This should help us indicate growth patterns and how strong each sample will grow. So other testing procedures are possible, and we've begun to discuss that, such as colony growth on a plate. We expect the more fits, Synechococcus 7942 to form colonies quicker, and so we could potentially use that as a measurement as well instead of absorbance. So we'll be looking into that as well. We expect substantial results to be around 7 to 8 weeks. And then after that point, it's expected to plateau unless we get some very large genetic change, which is possible, but it's random. That's why we have multiple populations in the hopes that one of the populations will hit the jackpot. So after the 7 to 8 weeks, we expect a plateau. But long term, hopefully, we expect to maximize our results and get a better Synechococcus 7942. Again, the potential for contamination is very possible. We're doing lots of inoculations multiple times over and over. So it's really important that we have these save points, which are really important. And even longer term, such as when the project is finished, we want to analyze the genetic changes that have occurred to each population and compare it to our wild type that we begun with, which we are still deciding on which wild type we'd like to use. But that's just some preliminary stuff. So onto our multi-cultivator. This is a really amazing machine that we have here. It's able to automatically grow these Synechococcus 7942 for us. It gives light, and it automatically can feed in gas for us. We can put in air or CO2, whatever we choose to use for our experiment. My experiment specifically will be using carbon dioxide. And then every 10 minutes or so, it'll automatically read the absorbance of the sample. So we can use this computer down here to track their growth over a period of 2 or 3 days. This machine is really amazing, and without it, this process would be almost too ridiculous to do. But luckily, we have this, and so we're very excited to keep using it. And now, to dive deeper into the future possibilities, because this is where it gets more complicated, the Pakrasi lab was really big in figuring out what genes could be changed to allow Synechococcus to grow quicker. These genes are the ATPA, PPNK, and RPAA genes. And all of that is right here on the right side. It's obviously hard to read, but basically it just goes through what's changed, and it compares it across different species of cyanobacteria. What we want to do is we're going to look for changes in these same genes, which we hope to achieve, and we also want to find different gene changes as well that also affected the growth of cyanobacteria. That way we can hopefully build on the knowledge of fast-growing Synechococcus. And then finally, here's a lot of our sources for what we did. The Linsky lab at Michigan State was really helpful in experimental design. Justin Unger's article over discussing comparative genomics of Synechococcus was extremely helpful. And then the Milo Johnson is the Pacarassi study, which was it's what we aspire to hopefully improve upon. And then, of course, special thanks to my research laboratory, the Burnap Lab at Oklahoma State, Dr. Burnap, Ross Walker, John Dorlon. These guys are helping me out, and I'm helping them out, and they've made my research experiment experience at Oklahoma State really amazing, and I'm really excited to be able to share this with you all, and also continue into the next few years. So, yeah, thank you.